Hi all, and welcome to the EVS program Capstone presentations. I know the setup may look a little bit different this year, but that shouldn't stop any of us from sharing our research with all of you. Today, I will be presenting my research that I conducted in the summer of 2019, titled Amendments of Lime and Mycorrhizae Improve First-Year Plant Success in an Experimental Planting on Abandoned Coal Mine Overburden in Appalachia. I know, quite the mouthful. I conducted my research on an abandoned coal mine overburden located above Horn Run in Marianna, Pennsylvania. This overburden is a result of the effects of coal mining. The process involves removing and dumping large quantities of waste material at these dump sites. The ground at these sites, which Dr. Kilgore and I often struggled to call soil, contains a high concentration of metals, very low nutrients, and has a very low pH. This makes it incredibly difficult for plants to survive as they draw many elements from the soil that are essential to their growth and overall health. Heavy metals in plants can have a detrimental effect on photosynthetic rate, chlorophyll content, transpiration rate, and water retention. Many plants will not survive under these conditions, but those that do will have affected levels of minerals, antioxidants, and of heavy metals in their leaves. These are not photos of our abandoned coal mine site, but of sites very similar, and you can see that not much grows at these sites and that the soil here is extremely dry and rocky. Studies have shown that some plants, such as little bluestem, can naturally survive on these materials, while others, such as annual ryegrass and lavender, are being studied as initial cover or as potential crops. Other studies are exploring soil treatments that could be effective in supporting vegetative recolonization of these sites, including a lime treatment, mycorrhizae fungi, and a straw covering. The goal of my study was to determine which soil amendment and plant combination can improve plant success on the overburden. This is a process known as phytoremediation. In the summer of 2018, Dr. Kilgore and WJ alum Lucy Elkin set up the experimental planting site. There are 60 experimental plots that run in 5 vertical lines of 12. This is an aerial view of the site, courtesy of Dr. March and his drone. The site is surrounded by deer fencing to prevent vandalism and herbivory, but between May and July, we realized we had a groundhog who was eating our test subjects, and additional steel wire fencing was added to prevent further herbivory. Each plot was randomly assigned one of 5 soil treatments: a control plot with no treatment, lime, mycorrhizae, a straw covering, or a mixture of all 3, and randomly assigned one of 4 plants: little bluestem, annual ryegrass, lavender, and strip-mine seed mix. Strip-mine seed mix was introduced to test the effectiveness of a commercial product designed specifically for areas such as our site. Each combination of plant and soil treatment is repeated 3 times throughout the experimental plot. The plants were all planted in 2018, and my research was focusing on monitoring their growth the following year. Plant height and plant density were measured to observe plant success. For plant height, I measured the tallest plant within the plot, and for plant density, I counted how many individuals were within a 10-centimeter metal ring that I placed in the center of the plot. I measured chlorophyll content, fluorescence, and photosynthetic light response as indicators of plant health using various technology. This data was recorded in both May and July of 2019. All plants measured for height in both May and July were significantly taller when the soil was treated with lime, mycorrhizae, or the combination of treatments. With the exception of little bluestem, plants that were treated with straw were significantly shorter than those treated with lime or mycorrhizae. Plants that were treated with mycorrhizae showed the highest mean chlorophyll content, but when treated with straw, the chlorophyll content was significantly lower than all other plant treatments. Results for the other testing factors, density, fluorescence, and light response, did not provide any significant results, but often lime or mycorrhizae performed better than the other soil treatments. This lack of significant data could be attributed to a low replication number per treatment, but as this testing site is only so big, this is a difficult factor to overcome. A similar study, conducted by Gray and his team of researchers, supports lime and its effectiveness in phytoremediation because of its ability to increase the soil pH and decrease metal availability. Kahn and his team support mycorrhizae fungi as a phytoremediator in their studies because mycorrhizal colonization at the roots of plants increases surface area and therefore nutrient acquisition. The fungus has also evolved a heavy metal tolerance, keeping these metals from being taken up by the plant. Continued monitoring will be essential to understanding how long-term effects of Lyme and mycorrhizae amendments on coal mine overburden. Thanks for watching.